Great, so I'm Adrian Isaacs, and I have a research lab at UCL, so just down the road a little bit, at the Institute of Neurology. And my lab has been going for several years now, and we're really very much focused on trying to understand the genes that cause FTD. And ultimately, the ultimate goal, I guess, of our research is if we can understand these genes that cause FTD and what goes wrong, ultimately that we can then hopefully intervene and start developing potential therapeutics that can correct what's going wrong with these genes. So, there we go. Just to give you a very quick outline of what I'll talk about, I'll very quickly talk a bit about FTD itself. Then I'll tell you about the genes that we know that cause FTD. And then I'll focus specifically on two genes that my lab works on, just to give you an idea about the types of approach that we take to try and understand these genes when they're discovered. And I'm going to speak for about 15 or 20 minutes, so not too long, so hopefully we'll just get enough to keep your interest. So the two genes I'll talk about, and I'll come back to this, are CHMP2B, that we call CHIMP2B, and another one that I've already talked to some of you about already, C9orf72. So I'm sure most of you are aware of this already, but frontotemporal dementia happens when people's brain cells are lost. So brain cells die in these two parts of the brain. So this is the front of the brain here, and here's the back. So when brain cells die, either in the frontal lobe, which is right at the front, or the temporal lobe here, then people will develop FTD. And depending where the brain cells are here that die, depends whether people with FTD end up having the characteristic changes. So we know if you have brain cells dying around here, then you're more likely to have what we call the changes in personality and behaviour. And some people have problems with language, and that can often be more affected with changes further back in the brain. And we call it a young onset dementia, which means it really occurs to people often, but not always, before the age of 65. And it's actually quite a common form of young onset dementia. So the most common form of dementia is Alzheimer's disease, and that's really common in old people. In older people it's really, really common, but it's also the most common form of dementia in younger people. But after Alzheimer's disease, FTD is the next most common. And I'd say we think there are probably about 5,000 to 10,000 people in the UK who have FTD. But of course, as I said, it's a little bit different from Alzheimer's disease, because in Alzheimer's you have these problems with memory being very prominent. In FTD, as I said, you can get more problems with changes in the personality and sometimes with language. But what's key for the work that my lab does is that about 40% or so of people with FTD, it runs in families. So when it runs in families, that gives us an opportunity then to find out what the gene is that causes FTD in that family. And this is a pie chart of the different genes that we know cause FTD. So I must say there's been really remarkable progress in about the last 10 years or so. So 10 years ago, we only knew about this one gene here, tau. And now you can see that almost 3/4 of FTD that runs in families, we know what the gene is. So many of you will have heard about these three main genes that cause FTD, so C9orf72, tau, and progranulin. So between them, they make up rather a lot of FTD that runs in families. But my particular lab focuses on two of these genes. So one is C9orf72, and the other is CHIMP2B. So CHIMP2B accounts for this sort of very thin sliver of FTD, but we think it's really important actually that we try and understand the rare causes of FTD as well as the common ones if we're truly going to understand and really beat the disease. The other gene that certainly will affect more people in this room is C9orf72. So this was only discovered about 3 years ago, but you can see it takes a really big chunk, about 1/4 of FTD that runs in families is due to this one gene. So there's been a large amount of work from many groups really trying to understand what this gene does. And again, we go to the brain to look at what's happening. So actually, I'm just going to go back. So that picture I just showed you actually is a zoom in of one brain cell, so one neuron, but we've done the colouring slightly different here. So actually in blue on that slide is going to be the central part where all the DNA is, and then outside of it is going to be the rest of the cell. So we've really zoomed in now on one brain cell. So this is the centre of the cell where all the DNA is kept. And in someone who doesn't have FTD, all you see is just where all the DNA is. But with someone who has FTD with C9orf72, we actually see something slightly different. So this is a clump of protein, and it's different to what's building up in the Danish family, but there's definitely these clumps of protein that appear in these neurons. And that's quite often what you see in people who have something wrong with their brain, is you get these big build-ups of different proteins. But what was unusual is that we also saw these green blobs, and they're made up of something else called RNA. So RNA really is like a precursor of protein. So in order for proteins to be made, you make the RNA first, and that ends up leading to these proteins being formed. So now we had this very clear question, and everyone who was looking at this wanted to answer the same question, which was, so what's really making these brain cells die? Is it these clumps of RNA here? And we know there are other diseases where clumps of RNA can kill cells, or is it these clumps of protein? And that's actually quite a difficult question to answer, because basically one leads to the other. As soon as you have RNA, because it's like a precursor, it ends up leading to these proteins. So my lab basically spent about 2 years trying to come up with these clever genetic ways where we could look at the RNA separately from the protein. And after about 2 years, we worked out how to do this. So once we had this RNA and this protein, again, we wanted to put it into a model organism and see whether they killed neurons. And this time, we didn't go with the mouse, we went with another model organism, the fruit fly. So I don't know how familiar you guys might be with a fruit fly, but the fruit fly is actually used an enormous amount in medical research. And it has a sort of long history in genetics research. So a very famous scientist called Thomas Hunt Morgan, just around the turn of in the 1900s, realised that flies might be very helpful for us to understand about genetics. And that's because basically they breed very quickly and they're quite cheap and easy to maintain. And so there's a huge amount of work since the early 1900s on flies, and we now know every single gene in the fruit fly. Even more importantly, these geneticists, these clever geneticists, have worked out ways that we can basically turn on or off any gene that we want in any part of the fruit fly. So we could turn a gene on or off just in the eye, or just in the wing, and in fact, we can turn a gene on or off actually just in the hair bristles on the back of the fly if we wanted to. We could turn a gene on or off just in those particular cells if we wanted to investigate them. So we did this in a number of ways, but what I'll show you is we turned on this C9orf72 RNA and protein just in the fly eye. And it's quite easy to look at the fly eye, but actually the reason we like looking at it is because basically it's full of neurons. So they're photoreceptor neurons, so light comes in and they basically transduce that into an electrical signal that's then read by the brain. So they're full of these photoreceptor neurons. So what we did with this C9orf72 RNA and protein just in the fly eye, and it's quite easy to look at the fly eye, but actually the reason we like looking at it is because basically it's full of neurons. So they're photoreceptor neurons, so light comes in and they basically transduce that into an electrical signal that's then read by the brain. So they're full of these photoreceptor neurons. So what we did was we took flies, and this is just a zoom in of the fly eye, and you can see it has this very regular structure. We then took our RNA that we'd made separately from the protein and we put it into the fly eye, and you can see it has absolutely no effect, so it's totally normal. So then we put the protein in. So I said we spent 2 years trying to work out how to do this, and then we see something completely different. So the fly eye, when we put in this protein, is completely degenerated. And I think that's telling us something quite fundamental here, that it looks like of those two things we saw building up in the brain, it's these proteins that seem to be killing the brain cells. So now we can go on and try and understand how that's happening. But just to give you a kind of idea, and this is my last slide, just to sort of end on a sort of hopeful note. This work, because it was quite a fundamental discovery, sort of working out what was going on here, we published this data in in a sort of very prestigious journal called Science, that's sort of the most visible scientific journal in the world, basically. So it's had very wide impact. And since we published it 8 months ago, we've had nearly 40,000 people have viewed this. So there's a huge amount of interest, actually, in trying to understand how these genes work. And there's many, many labs in the world, my lab, many labs in the UK, and across Europe and the US, who are really focusing now on trying to understand these genes. And I hope, undoubtedly, as we understand more, we will soon be able to start developing therapies based on this type of knowledge. So I'll finish there and take questions.